Hi class, this is Nelson Jones here, and we are on our last group advanced concepts presentation, talking about our whole poster we have built over the past few weeks. Here is mine, and essentially we're going to kind of go through this poster and talk about ATP production, as well as how nucleotides and ribose are made and how they come together to make ATP that is used for DNA replication. So let's first talk about ATP production through cellular respiration, where 1-glucose is broken down to create 30 ATP through the process of cellular respiration. The 3 stages of cellular respiration, as we know, are glycolysis, the citric acid cycle, and the electron transport chain. We can kind of see all of these steps here on 6, 7, 8, and 11 and 12. And in glycolysis, we convert glucose into pyruvate, which results in a net of 2 ATP and 2 NADH. So here is our glycolysis, what we've created in pyruvate. And then from there, the pyruvate is then converted into acetyl-CoA, which it also creates NADH. And then in the citric acid cycle, we create 6 NADH and 2 FADH2s and 2 ATP because we have 2 of these acetyl-CoAs coming into the citric acid cycle, so we double them. And then in the citric acid cycle, and then we move to the electron transport chain over here on number 11. And electrons from NADH are donated to complex 1, and from FADH2, they're donated to complex 2, and then they're passed down the chain. And this pumps protons into the intermembrane space from the mitochondrial matrix, which creates the concentration gradient within the intermembrane space, which allows ATP synthase to create 2.5 ATP per NADH molecule and 1.5 ATP per FADH2 molecules, which results in 30 ATP being generated in cellular respiration. So this is ATP synthase and how those protons move from the inner mitochondrial space into the matrix. And as they do that, it creates ATP. And next, thinking about nucleotides and the sugars associated with that, we will go back to the first step of glycolysis and talk about the pentose phosphate pathway. And the pentose phosphate pathway is a metabolic pathway parallel to glycolysis. It uses glucose 6-phosphate, which is here, which is the first step for converting glucose to glucose 6-phosphate. And it uses glucose 6-phosphate to generate NADPH and pentoses, as well as ribose 5-phosphate, which is a precursor and the sugar that is used for the synthesis of nucleotides. And so if we move up to number 1 here on the poster, we have our glucose 6-phosphate. And essentially, it's a parallel pathway to talking more about the pentose phosphate pathway real quick. Essentially, it is a parallel pathway to glycolysis that results in the production of the sugars that make up DNA and RNA. And in the pentose phosphate pathway, no ATP is used or produced. And once in the pentose phosphate pathway, once the ribose 5-phosphate has been created, it is then reacted. Here we go, ribose 5-phosphate is then reacted with ATP to form phosphoribosyl pyrophosphate, or for short PRPP, which is here. That's a pretty key molecule. And PRPP is very important as it is used in de novo nucleotide synthesis in both the purine pathway to produce adenine and guanine, and the pyrimidine pathway to form cytosine, uracil, and thymine. And so we're kind of jumping across to pages 4 and 5 over here in the top right corner. You can see our PRPP here. And now we're looking at pyrimidine synthesis, which form sorry, we're going to start with purine synthesis actually first. So with regards to purine synthesis, we start with PRPP, and we have a 9-step reaction that results in the formation of I have trouble with this one, inosinate. And looking at page 5, we can see after this 9-step reaction, we have inosinate. We take inosinate here, and we then use that to form either ATP or AGTP, or GTP. And you can see the reactions here, the 2 different pathways that inosinate takes to form ATP or GTP. And then looking at pyrimidine synthesis, which I mentioned forms cytosine, uracil, and thymine nucleotides, we react bicarbonate with ATP to ultimately form orotate right here. So we're kind of going from here to our orotate, and it's reacted with PRPP and is then moved through a multi-step reaction to form urulidate, which is here, and then here, uridylate. And then with the uridylate, we can react to form UTP, CTP, or TPP. So you can see how those work and form those nucleotides. And so wrapping up with a quick recap, we'll kind of zoom out here on our poster. Wrapping up with a quick recap, we take a glucose from glycolysis and convert it to glucose 6-phosphate, and it enters the pentose phosphate pathway. Ribose 5-phosphate is a result of the pentose phosphate pathway, and it is a sugar that is used in DNA and RNA formation. And again, there's our ribose 5-phosphate over here. And then the pentose phosphate pathway also forms PRPP up top left there, which is a key for de novo nucleotide synthesis. And there are 2 ways in which nucleotides are made, the purine and pyrimidine pathways that we talked about over on numbers 4 and 5 of our worksheets. So this is my whole worksheet again, or poster, and I hope this was informative to everyone, and I hope everyone has a great end of the semester, and I had a great time working with everybody. Okay, bye.